Now I'd like to introduce one of my great friends. One of the wonderful things about putting this symposium together was that it was very easy for us to find a team of killer women working on ovarian research. and I'm very happy to introduce Angie Belcher. She's a great friend of mine and a longtime collaborator. A little bit about Angie. She's the W.M. Keck Professor of Energy and Material Science and Biological Engineering, and a Koch Institute integrative cancer researcher. She's one of the leading scientists in the field of nanotechnology, and she looks to the natural world for clues and inspiration to create extraordinary materials. Angie alters the DNA in bacterial viruses, which are benign to humans, but can create materials for practical human use. Her groundbreaking work has resulted in self-assembled materials that may be used as components in electronic devices such as batteries and sensing devices, solar cells, and display screens. She's currently researching ways to use her technology for purification of water and the early detection of ovarian cancer, which she's going to tell us about today. And for her inventions, she has won the 2013 Lemelson MIT Prize, which is one of the highest honors in innovation that we have. So I'd like to introduce Angie. Thank you.
Well, it's a huge honor to be here today to speak to this group and tell you a little bit about what we're thinking about and working and how it relates to chicken wire here. So, as Paula said, I've spent most of my career working in solar cells and batteries and electronics and working things at the nanoscale. And most everyone has heard of nanoscience and nanoscale, that it's a billionth of a meter, so it's really small. It's 100,000 times smaller than a human hair or the thickness of paper. And my kids always want smaller iPhones, iPads, although now they want bigger iPhones. But something that's special about nano is it's small, but that's not the only thing that's special about nano. When you take just a collection of atoms and put them together, a lot of special things happen. They change their optical properties, the way they interact with light. They change their electrical properties, and a lot of wonderful things happen. And so Tyler, a couple of years ago, asked me to come work on cancer, and I said, wow, yeah, I work on solar cells and batteries. But I was very excited to be a part of it and decided to come over and think about what I've done in my earlier career and how that might be useful in cancer research. And it really came back to nanoscience. And people might have heard of this material called graphene, which is a single layer of carbon, and it's a very special material. It has a lot of interesting properties, and something called carbon nanotubes, which is basically taking, so this chicken wire here, you can think of it as a molecular layer of carbon. And it's on the atomic scale, or when it is put together, becomes on the nanoscale. Well, you can take this basically chicken wire, this carbon, and roll it up, and it becomes something called a carbon nanotube, which actually has a lot of interesting properties. But one of its interesting properties is that it is fluorescent, and it can be colored. And it gives off an interesting wavelength of light. This would be a carbon nanotube right here. And so it's 1 nanometer in thickness, about the thickness of DNA. And basically how thick it is and how it's turned when it gets rolled gives it different fluorescent properties. So you can think about these carbon nanotubes having different colors. And you think about these carbon nanotubes as being beacons. My students made this for me today. This carbon nanotube that's lighting up. Like here, now you can think of it actually as just being one particular color of light, and lots of different carbon nanotubes that are different colors of, give off different colors of light. So you can think of it as a molecular beacon, but a nanoscale molecular beacon that we can use, as Dr. Beer was saying, to try to detect and find very small ovarian tumors, with the idea of the margins, and can you find tumors that are deeply buried inside that surgeons, when they're going in to remove the tumors, wouldn't normally see. And so this is the same material that we use to make batteries. And when this battery was famous, it went to the White House to meet President Obama. I wasn't invited, but my battery was invited. And solar cells, but it's also a useful material for imaging cancer as well. And so it was also mentioned that we work on bacteriophages. These are a structure. You have you're full of bacteriophages, and they're everywhere. But one thing that's nice about them is they have DNA, and so it's actually single-stranded DNA, and it has just a couple of genes. These different colors represent different genes, and these genes code for the proteins that make them up. So this bacteriophage is a wonderful organism, and it is actually about the same length as these carbon nanotubes. And by going in and manipulating these DNA sequences, let me see if I can get this to work, we can manipulate, there we go, we can manipulate different proteins. So imagine these proteins on the tip here, imagine their job is to find cancer cells and bind to them. Okay, so we can engineer these to have certain protein recognition sites that bind certain kinds of cancer cells and bind to the cells. Now imagine that we engineer these proteins here to bind to carbon nanotubes, our little molecular beacons that give off particular wavelength of light, and you bind them to the structure. Now you have this basically biological functioning unit that goes in, finds tumor cells, binds to them, and gives off light of a particular wavelength. Now you can use a very small amount of this, and you can inject it IP to be able to look for, find tumors, maybe eventually tell you information about what kind of ovarian tumor it is, and give off light. So that's what we've been doing. And so we're new to working on ovarian cancer, and our first paper just came out this week in collaboration with Professor Beer's group and Professor Sangeeta Bhattia's group on using these to find very small tumors in mice. And so these are tumors that are less than a millimeter in size, and we've shown that it's useful to basically deliver this IP. An imaging system didn't exist in order to do this, so we had to develop the imaging system to go with the probe. And so my great team of engineers and scientists spent a lot of time developing a new imaging system that we could use for starting in mice to be able to look for these tiny tumors. And then working in collaboration with Lincoln Labs as well, they developed the software to get information about when the surgeon goes in and basically shines light, and the tumors glow, and the surgeon goes and removes them, but also to get information about those tumors. You know, how deep is it? What's its total size? And eventually, we'd like to get molecular information about that tumor so that the physician and the surgeon can have them in real time. So that's what our project is. That's how you go from solar cells and batteries to thinking about ways of finding and scoping out teeny tumors. Now, one thing I didn't mention is that you're going, well, you know, I know that the visible light that I interact with every day, I can't shine that and have it go all the way through my body. And you know that there's different wavelengths of light. These are samples that have the optical density of breast tissue. And I have two different laser pointers here, a green laser pointer and a red laser pointer. Now, if I take this green laser pointer and I shine it at this, you can come do this later if you want to. You see a lot of scattering, a lot of reflection. So that's not very useful. You can't really get deep inside of the body. But as you go to this red laser pointer, you're actually able to go deeper into these tissues. And my students have been working on using something called near-infrared light, so even longer wavelength of infrared light to be able to go all the way to 9 or 10 centimeters in depth just by shining light and be able to get information out about where a tumor is. And so you say, well, what is 9 centimeters? Well, this about here is the distance of 12 centimeters. So I think we're at, as of yesterday, 10 centimeters. My students are in the group working every day to try to push it. So you could actually think about having light go in and be able to see something this deep inside the body without having to use something more expensive like an MRI or something that gives you lower resolution. But this could also eventually be less expensive. So where is it? It's in the basic science stage. We've been developing the probes, we've been developing the equipment, and we've been working in mouse models on it. But it's something that it's our way as an engineer of looking at some of the current limitations in imaging. So these are carbon nanotubes made out of carbon. These are my examples of other kinds of nanomaterials that will allow us to eventually even get deeper into the body. So you can think about engineering these to bind to particular cells and then carry with it different nanoparticles that could also act as little molecular beacons and give off different signals to give you information about it. But you can also imagine this is a drug molecule. So if you imagine binding this giving off light, and then eventually being able to deliver a drug as well. Again, it's all at the very basic science stage. And I have a couple of pictures I might run through on this. So this is a fantastic team from MIT, Koch, MGH, and MIT's Lincoln Labs. Why were we interested in doing this? Well, we were interested in getting something that actually could go very deep and be non-radioactive, inexpensive, and quick. This is a special window. It's called the second window near IR, and this is the wavelength here between 950 and 650 nanometers. And it just happens to be that certain structures of the carbon nanotubes are perfect for this wavelength, or I can engineer the structure of nanoparticles to be perfect to sit in that wavelength. Why hasn't it been done before? Well, a lot of probes didn't exist in this region. So as engineers and as scientists, we're in there thinking, can we develop better probes so that we can be able to image in this wavelength? And we would like it to be one day where it's inexpensive enough that you could use it in your physician's office instead of having to go into specialized facility. And so we've been, along with the team that I mentioned, we've been developing the instrumentation, developing the probes, and trying to use it as both a diagnostic method and a surgical guided tool. The thing is that we can engineer this to bind to anything we want. It can be different kinds of ovarian cancer tumors. It can be breast cancer tumors. So this is very engineerable. This is very engineerable to stick to different things we want as well. And you can think about, do I need this whole structure? Maybe not. Maybe I can just use this protein here along with this nanoparticle here, which is also an effective way of doing it. So here's some data from our paper that just came out in Proceedings of the National Academy of Sciences. And one of the things that's nice about this particular light region is that there's not a lot of background. And so if you're looking for something and you get a lot of extra light background coming that you're not interested in, it makes things murky and it confuses things. But because at this wavelength things are transparent to blood and tissue, you can look deeper and you can get more information out. And so this is actually what a mouse looks like that has ovarian tumor, but hasn't been injected with our nanoparticle cocktail. And you don't see any background fluorescence. That's really nice. That gives you hope that when you have signal, that the signal is going to be a real signal. Now this is pre-surgery. This is a mouse that had a high burden of ovarian tumors. And Sangeeta's group helped us pick that model and did all the work for that. And then we delivered our cocktail into the intraperitoneal space, and now you get a map of where the tumors are. And so working with Dr. Beer's group, first of all, his surgeon went in and did surgery on a mouse and removed all the tumors he could see by eye. He didn't look at our map. He removed all the tumors he could see by eye, and then we took a picture of it. And he's a great surgeon, but he hadn't removed all the tumors. But now we give him the map, and he goes back in, and he can find many more tumors that he couldn't see before. And so our first generation instrument, the one that we just published, was based on this map. You provide a map on the computer that has the information that helps you decide where it is. Our third and fourth generation instruments are real time. In real time, where there's a surgery going on and you're looking at the screen and getting information about it. Oh, it's buried down here. It's about this size. Again, it's all just in mice. But using this, even just using the map, he was able to find tumors that are less than a millimeter or smaller. Now with CT imaging, the gold standard is 1 centimeter, and so this is much, much smaller. And so it's hopeful that we'll be able to use this technologies and parallel technologies to get more information. Here's an instrument that's actually up in our labs that's working right now. And using special software, we can start to pull out more information. And this is the work with Lincoln Labs that they've been able to pull out more information to provide the physician real time with information about depth and size, and even break it up saying these are the larger tumors, meaning above 2 millimeters, these are the medium, and here's the small ones. Here's a map of where they all are. Not where they all are, but where a lot of them are. And so far in the tumors that we've removed, we've tested them using traditional histology, and 49 out of 50 of them were positive for the type of ovarian cancer that we thought. So this is This is early stages. This is an engineer's or a collection of engineers' approach to thinking about ovarian cancer, and it's been a huge privilege to work with the team here, and I hope that we'll continue to make more progress in pushing the envelope and what is possible for finding tiny tumors. So thank you.
Excellent. So again, we have a microphone that will be passed to people who have questions. Any questions from the audience? This one right here.
Hi, thank you for this very interesting talk. I was wondering, when you were showing the number of nodules that you removed, the one that were less than 1 millimeters were higher than unguided, but for the other that were more than 1 millimeter, I don't know if I missed some information.
No.
So does it lose sensitivity, or is the marker an early stage marker of the cancer, and so when it gets bigger it's like hidden or something?
No, that's a great question, and I just went through that really quickly. But he was able to see those tumors with his eyes and able to remove them. So the spike comes from the fact that those are tumors that he couldn't see by eye. He removed the tumors that were in between. Yeah, but that is an interesting question. It does give me ideas of things to think about to differentiate those. Yep, thank you.
Here.
Was that with the eye or with something like the Da Vinci robot? Just with the eye?
Okay, so the initial data that I showed, the surgery that he did was by eye. And the improved was when he still did the surgery by eye, but followed it with a map on the computer screen of where we thought the tumors were. Now the next generation instrument is all still done by eye, but then looking at it simultaneously, going back and forth, looking at it simultaneously on a computer screen in real time. So it does rely on the skill of the individual.
Great. We have a question here.
Very nice result. I'm wondering how susceptible these tumors are to thermal therapies, and if you can use your imaging technique to now go ahead and hit those tumors, maybe that residual nodules that the surgeon cannot see, to go ahead and take advantage of the properties of the carbon nanotubes and kill them by heat.
Yeah. So thank you for that question. We have actually done those experiments. And you can talk to my students. You can raise your hands, guys, that have tried those experiments in the lab, sitting right next to you conveniently. You can talk to them afterwards about that. But basically, we have had that idea of either heating the carbon nanotubes by themselves, attached to the tumors, or using gold or other metals, and that's in our toolbox of the things that we're interested in. Right now, we've really had to spend our time focusing on developing the probes and the imaging instruments. And yes, thank you.
Yes, I have a question right here.
How long before we'll see it, being used in humans? How long do you think that'll take?
Well, I don't think we're even close to testing in humans right now. Like I said, our first paper came out this week in mice. There's a lot of questions that have to be answered in animal models first before we can think about human trials. Of course, everything that we do in this building has the eye of, if it's basic science, it's basically towards translational medicine. So if it wasn't something that we thought had applications in humans, we wouldn't be working on it.
All right. Any additional? All right, we're all set. Let's thank Angie one more time.